Okay.
Good. Yes. Sounds good. Okay. Okay. Thank you. I welcome you all. And today we have Professor Rahul Roy from Chemical Engineering Department. And as I know him, he is doing work in microscopy and trying to understand diseases. Okay. So I'll go a little bit about his studies and all. So Professor Rahul Roy earned his PhD degree in biophysics and computational biology from University of Illinois, Urbana-Champaign in 2007, which is considered as a epicenter of biophysics. So after a postdoctoral stint at Harvard, he joined the Chemical Engineering Department, Institute as assistant professor in 2012, and subsequently now he is holding the position of associate professor. And also he is associated with other departments like Center for Bioscience, Biosystem Science and Engineering. His lab is quite interdisciplinary, and his lab is engaged in developing new technologies which use single molecule detection, quantitative genomics, and high resolution. And his team has invented and come up with several assays and technologies to help understand and manage infectious diseases over the last 10 years. And mostly the idea is that how to use single molecule techniques or microscopy techniques to understand it. And this is where I think Rahul is excelling now. And I'm really impressed by the first slide. So I'm just looking forward to listen to this great talk. So Rahul, can you please stay?
All right, everybody can hear me? Can hear me, I presume. And I don't know whether it's the best place to stand, so I will try to stand on that side so that they can see you, and if they have questions for me, somebody will hopefully tell me, right?
Yeah.
So, okay, so first I have a question for you. How many of you have heard of superresolution spectroscopy? Somebody has heard of superresolution spectroscopy? How many people know about fluorescence imaging, fluorescence-based imaging? Okay. Have you done any imaging on your own? No. So, what is your background? Engineering? How many engineers do you have? Engineers. When you say engineers, you are biotech engineers or electrical engineers? Mechanical engineers. Okay. All right. How many biologists? Mostly biologists. The rest are what's the class? Huh? Physics, how many? Okay, and what about you guys? You are in Arthur's lab, all of you. All right, so now I have to reorient myself a little bit. So what we are going to see today is using imaging and a particular kind of imaging which has some fault to understand systems which are complex, and most of the biological systems are complex. So I'm not going to tell you about all the biology that we do, but I will tell you how we have been using and we can easily implement any system, any complex system, and it doesn't need to be biology. Okay? You can come up with a system which is complex enough. You will see some complexity today, why we use what we use, and those are these single molecule experiments. You have already heard talks, if you have followed this series about super-resolution microscopy. People use single molecule imaging to get super-resolution microscopy. But because we have already heard about super-resolution microscopy, I didn't want to again kind of give you the same kind of presentations what we have. So today we are going to learn some biology, but forget about biology. You probably never will hear about these biological processes. Think about how this techniques allows you to investigate complex systems. Okay? So that's the theme. Okay. Partha already introduced me, so I don't need to worry about introducing myself. Let me try to make sure that I can move my slides, which is not happening. Okay, there you go. So today I will tell you how do you image single molecules. Okay, actually single dyes which are emitting light. Okay, single molecule. Okay, and because we are going to be imaging single molecules, a lot of things are possible which are not possible. Okay, and why would you like to go single molecule at single molecule level? What do you need to do single molecule imaging? And so you will see a lot of those. I'll tell you about some glimpses of. They are not. I can have a lecture on all of these individually, but today I will just tell you about briefly how it works, and if you're interested in more, you can come up to me and help them. Okay, so some of the techniques I will introduce and what it allows you. All right. The reason I use single molecule techniques is because I see biological systems like an orchestra. All right. Imagine a cell, and inside the cell there are several molecules that are present. Each molecule has a role to play.
You can keep this with you because audio is a little low. Okay. All right. Shall I place it here?
Yeah, fine. All right.
So I have different types of molecules inside the same cell. Each has a role to play, right? But they don't play all the time. If you have an orchestra, they do their job at certain times, taking cues from certain others, right? There also might be a master regulator who might be controlling the orchestra, certain set of molecules which job is to instructs everybody else. If I were far away and just listening to the music, I would definitely not be able to tell them. Biologists do the experiment even worse than that. They will take many orchestras, put them together, and then they will ask, what music do I hear? What do I understand from it? Yeah, they are clever, of course. Very clever. They will take out one orchestra piece and then say, what happened to this? Or they will put more of those pieces and they will ask what happened to the cell. If you are a biologist, you have now understand what happened. Or you take out the orchestra in the context of their seating arrangement, their violence and stuff, put them in a soup and then ask, hey, inside the city, how do they play? And so all that tells you that that's not the best way to measure and understand their functions. It's a complex system. You need to be able to interrogate them at a level which will give you more detail. And what I am trying to argue is that instead of taking them out of the context and just purifying all these folks and trying to see what they are doing, that's also not true. You want to see that person in their context. And this is biologically extremely true for many scenarios. But this is something that I would like to show you is a cartoon, of course, of a small fraction, about 1/16th of a part of a bacteria. And you can see all sorts of polymers in there if you don't care about the biology, but these are DNA molecules here, they are DNA binding proteins here, they are these big ones are ribosomes that are taking RNA that has been generated from here, making proteins out of it. This is a membrane on which you have tons of other things, right? Each have a role to play. And you will notice the I'm going to try this, and if there's a problem, please let me know, then please you will. Okay, so look at the dimensions. Okay, so there is no microscope. You have to get it in a microscope. The fraction microscope will allow you. Right? You get into EM, but then you will only get a static picture. One of the orchestra is in listening to the music, the function that just. Okay, so this is going to be a problem if you're just going to take snapshots in an EM. Right? I can do that. Oops. All right. Now, the consequence of having so many molecules, each at different kind of a polymer, they also can have their own modifications. If you hear about them, these are just chemical moieties, so they can have their own modifications. And the consequence of that modification is that even though the average is a broad average, each of them have slightly different activities.
Okay, you can have static differences. For example, here is a molecule here which has a certain level of activity and some a certain number of molecules that have much higher activity. You can look at two trajectories of single molecules. They have two states. They are flipping between two states in equilibrium. But you can realize that the kinetic rate of this one switching back and forth between the two states is much faster. It's the same molecule, according to me. Okay, it's the same chemical molecule, but it has completely different. And this has been found again and again and again when people have started investigating. Okay, there is another advantage. Right? Every conformation, every function will have a spectrum of intermediates, and you will only capture a bulk, even if you have triggered the reaction at one point of time. That's what biologists do. They trigger the reaction and then say, okay, what happened over function of time? They measure some average state. And what you get is actually the average of many, many different species giving you one average. You can be clever about it. You can have a simple system where you can interpret it. In other cases, you can't. If I had been watching this molecule only, then I would have seen this go through all these transitions. Right? So I need methods to be able to do that, of course, and that's one of the effects of the problem. Here's another very simple example. Here's a molecule that goes from this state to this state via an intermediate. Okay, the intermediate could be a very critical intermediate. If you don't form that intermediate, you will never form the final state. And of course, if you had taken an average signal, then this is what you do. Okay. You would have completely missed that. All right. So what I'm trying to convince you is that these molecules themselves are different. Here's a very elegant experiment that was done, taking one enzyme. We haven't heard about enzymes. These are catalysts, biological. You put them inside cell, they will do their job. You take them out, they will do also their job. You can measure their activities, and somebody measures the activity of a single catalyst as a function. What you see is this below this dashed line is background, but above this you will see the activity of the enzyme as a function of time. And what you notice is that the enzyme, if you have been taking any biochemistry classes or catalysts, you every time model them with a microwave equation, and you say, oh, the activity is going to saturate and give me continuous turnover. You don't consider any heterogeneity in the activity it will show. Look at how different they are. For some periods of time, it's showing almost 3 times the activity. Why? Well, it's a polymer, remember? So this polymer is fluctuating in the thermal path. It is every time taking all sorts of configurations that its right changes and hence the activity. Okay. For some cases, you might not want to know anything apart from the average rate. That will work fine. But inside the cell, I would argue that this becomes. Why? Well, inside the cell, so this is some numbers for bacteria. Inside the bacteria, you can have on an average about 10 molecules. That's it, of a particular time. That's it. Okay. Yes, there are some proteins which are necessary. They are present in high copy numbers, 100 copies, 1000 copies, but that's it. The bacteria is already very, very small. And some proteins are present, about 30% of the proteins are present at 1 copy or less. What does 1 copy or less mean? One bacteria has 1 protein, or sometimes the bacteria has no protein. Okay? So that bacteria has no memory that it is supposed to do something because it doesn't have the protein, it will behave completely. Okay? And the numbers are not dramatically different, maybe a little higher in yeast and higher organisms, but biology, in sense, is happening in these very low numbers. And hence, the properties of a single molecule. So if you take a pocket full of enzymes, you don't matter, it doesn't matter. But here, it actually might matter. And we have been finding this again. Okay. All right. And the consequence of this is that you cannot treat systems, the macro systems that you normally are used to, mechanical systems, electrical systems, where the number of molecules doing this exercise is very, very large. You can't treat biology the same. Okay. Here's an example of a reaction where the numbers of molecules is small, and the consequence of it, I'm not going into the details, is that under the exact same solution conditions, you have bacteria here which is showing very small numbers. It's not as if these are dead bacteria. They were producing very small number of proteins, and here is another bacteria in the same solution, right in the center producing something. We find them again. Okay, so heterogeneity becomes extremely large, and the consequence in this case, I know for a fact, is because of these small numbers and some genetic circuitry that controls it. There's some feedback in that, so that's at the level of how you impart control inside. Okay, all right. You look at any cell, and you will find the gene gets turned on and off all the time because of these small numbers of transcription which control the production. So think of it as a tap, but instead of opening the tap and letting it open, if you put water all the time, the tap is being opened and closed. Consequences, you get proteins being produced from the mRNA, but they will have a decay. The next time the gene turns on again, you will get a burst, and then that's more common. Of course, this is a schematic. I can show you that data also. All right. And that's again the consequence of biology happening in the universe. And this is why we think measuring single molecules is the best way to understand this. Otherwise, I will not be able to interpret this in any other way. Okay, you have to make sure that you treat all the biological systems as together. All right. So now that I have convinced you that the single molecule measurements will help you in interpreting the data, I'll tell you about how to do single molecule measurements. And my kind of talisman for understanding and imaging single molecules is in these 4 key concepts. Okay? I'll tell them in detail. You need what is called well-behaved protocols. I will tell you what you mean by well-behaved protocols. You need what is called background suppression, and you need high quality, high numerical aperture optics and sensitive detectors, and you need some strategies. I call them distribute and immobilize strategies. To understand all these ideas. Okay, so fluorophores, these are small molecules that you will tag the internal kind of state of the cell. Also has some small molecules. These small molecules are fluorescent to a certain extent, but they don't give you sufficient photons that you can kind of track them in the image. So what we normally do is we take fluorophores, which themselves are very bright, we put them on molecules of our interest and track them. Okay, if it is a protein, we can tag it with a small organic dye, or a fusion protein with a green fluorescent protein variant. Some protein which itself is fluorescent. We can genetically tag that. Okay. If it is a small molecule like a peptide, you can put it on an organic dye. If it is a large molecule, you can put a quantum dot and so on. All of these are examples of good dye molecules. You say, can I track ions? Not this way. Fluorescent molecules that will be much larger than the ion that you are interested in. You can't do it. All right. So, what do you want? They want them to be bright. Some very large extinction coefficient, good quantum yields, right? And then you want them to be photostable, which basically means that they should not undergo photophysical changes, aggregations, chemical changes, because that will change their intensities, and that is not what you want. You want steady levels of fluorescence from them, right? And they should have large number of photons that they should give out in a very short period of span, so that you can detect it using your detectors. And they should be water soluble and small. That's because you want to contribute them to a molecule of interest. And if you are not water soluble, then in the milieu of the cell, they will aggregate somewhat. Okay, and there's a list. This I'm happy to share on this list if you are interested. But this is also present in paper. I will. I'll not go through the list. Okay. Now you need to understand some degree of how the single molecule emits light. And the way it does it is by getting excited when you shine a light on it. You get it excited to an excited state. In this excited state, either S1 or S2, it will kind of collapse down by some vibrational energy that is released to this kind of a state. And from here, the ring come back to this ground state. When it comes back to its ground state, that's when it releases its form. Okay. It can also do other sorts of processes. So the fluorescence is always competing with other processes, including transitions to filter states, which usually are dark. when it releases the photon, this is when you see the photon. If it goes here and then comes back, you might not actually see a photon of the wavelength. Okay, so lifetime is the time it takes for it to come back to its ground state. That's the time it took. But of the order of a few nanoseconds for good dyes. Saturation is how many times can I do this before I can't go any more, I can't get any more photons. And pumping it down and down and again and again and again. Beyond a certain limit, if it's 1 nanoseconds, then I can get only so many things within 1 microsecond. Right? You at least need to give 1 nanosecond. So there will be all sorts of dark states. You don't want that dye molecule that goes to dark states to be made of times. And of course, you want to avoid lots of fluctuations in intensities. So avoid situations where it goes to S2s and S3s and filtered states and has many different intensities. Its conformation can also change and can give you a completely different emission. Okay, so those are things that you look out for when you pick. So you can't pick any dye and start doing experiments. You will see that molecule, but it might give you many other complications which will take interpretation of the data. All right. You can also increase photostability by other methods. We use standard techniques like, so the reason we use these is there are always other molecules inside the solution that will kind of chemically react to the molecule and photobleach them, take them away from activity and giving photons. So these are enzyme cocktails that have been already established. You can also remove oxygen from the system because oxygen has these free radicals. Oxygen radicals react with time very. They are highly. They will photochemically free. And you can, people have shown that you can directly take some of these stabilizers, small molecules, and put them directly on the dye, and then that also. If you are interested in learning more about it, and some of them are in the review that I did. Okay, this is the comparison of the dyes. Depending on which dye you pick, you can get larger or smaller photostabilities. And in presence of the molecules, you can increase the photostability further. So the numbers are not so critical, just that you can do this for a very large scale. So these are tricks of the trade that people pick up. But you can do them, and I'm just telling you that how people do this. Okay. You also need to tag them, right? And there are many ways of tagging them. So again, this is a list. It doesn't really matter. You can take advantage of the fact that you can have reactive groups like on the amino acids, you can have an amine group like lysine, arginine that will react with a chemical reactive group, like very well established reactions in chemistry that you can take advantage of. You can take thiol groups like cysteines and react it with melamine groups. And you can buy the dyes which come with these groups. So you can tag any molecule to your liking. If you have one cysteine, then you will have one dye. Okay, so you can take advantage of that. Genetic tagging I mentioned, you can encode in the protein itself the sequence of the GSP protein, the green fluorescent. The protein will be produced, and right after, with a linker on it, it will produce a green fluorescent protein, and that protein will emit the light while the first protein will do its job. Okay, and of course, people when they are doing superposition imaging, they would use antibodies because they are antibodies against many proteins. They will label the antibodies, and there are some other tricks like I've heard about them. Okay, then there you have to worry about which chemistry to use, where to place the dye. Sometimes it might even matter. Okay, again, too many details to give you today. So if you are interested in specific questions, please ask me later, or we can individually. There are many illumination methods, all targeted, and when I see these plus points, they are better in their ability to cancel out the background that is going to come from the rest of the solution. Why? Well, why do I see the moon only in the night? It gives out less light. Single molecule will give out less light. If you have other sources of background, then you won't see. Okay? So you take advantage of the fact that you can indeed reduce background, and that reduced background can give you now better ability to see your signal. Epifluorescence is good for only certain kinds of I'm not going to detail too much about it. These are different methods. I will show you some examples of how they are accomplished, but I have kind of ranked them on how much background separation you get. And hence you will find these methods are quite commonly used and not so much of it. If you're not sure about what these are, again, you can look up. Okay, here's a very simple diagram for showing you how a setup like this would look like. So for fluorescence, the three key things that I told you about is you need to be able to detect. You need a good camera. I will show you some examples of it. You need a high NA objective. You need some light source which is narrowly distributed in terms of the excitation. You can control it using some controllers. If you don't know the details, don't worry about them. The point is, you can also illuminate it in different ways. Okay, right here, the lens here is focusing it at the back focal plane of this objective. And when you focus it at the back focal plane and put it right in the center, then this will go up like this. This is the peak fluorescence. It illuminates literally everything that. Okay, not only in the plane right from the surface all the way. Everything gets illuminated. Completely radical way of illuminating it is instead of putting it right in the middle, put it at the side. When you move this thing to the side, what will happen? It will start bending, right? And if you bend this light, a lot of excitation out here has been removed, and instead you are kind of illuminating a very small region, and it is also going in the sideways. Doesn't give you so background. We call it highly inclined illumination. That's commonly used for illuminating cells. The cells are about a few microns thick, so this gives you very good contrast. But the extreme versions like curve, where what you do is you keep going this way, sidewise from the center, and then at some point of time, the light will start doing total internal reflection, come back. You have done maybe 12 standard number of these total internal reflection. Okay, when it comes back, however, there's a little bit of a light that leaks through because of surface plasmons to the other side, but it has a very large decaying field. It very quickly drops. So it illuminates only things which are right in the vicinity of the person. So if you have some molecules, let's say on the membrane, which is sitting right on the surface, you'll see some examples of it. We use this technique a lot. Okay. For everything else, you need at least these things. Good excitation, some laser sources, because you need some high power. This is not just getting some light. You're asking one molecule to give you sufficient. Okay, so that you can see it. So this is what you need. Some good objectives, because you don't need to just collect light. You need to collect almost every light that is emitted. Right? So usually this is done by high magnification objectives because they have high numerical aperture. Numerical aperture corresponds to the ability of the objective to collect light from a large distance. You want high transmissions in almost all visible range. If you buy a high-end objective, you will get all these problems. Okay, so look for these numbers if you are looking on the objective, whether it can do it or not. You need filters. Good filters basically will prevent, remember, background suppression. I want to remove everything else but look at the light. You need high transmission for your wavelength and should block out everything to the OD of 10 to the power 6. Right? Block out everything. Right? You want achromats so that you can remove chromatic aberrations. If you're not sure what terms I'm speaking, please speak to us. And you want high transmissions in the wavelength. All right, so here's what I was talking about. You can see that this has transmission, very high transmission. This is the filter that you would use, a high transmission in excitation. So this is for making sure everything else is blocked, but only allows excitation in this. So the blue one is an excitation filter. This would have an emission filter. So this will tell you the light it wants to see, fluorescent light it wants to see. And this is a dichroid that is mirror that was sitting at a 45-degree angle that was allowing the excitation light to go in. So if you didn't notice that, yes, this is the dichroid molecule mirror. The dichroid mirror basically allows the excitation, that is the blue light, to come in and gets reflected. So it is a special mirror but it will transmit everything which is longer wavelengths. It is usually super fluorescence. And this is how you ensure in this kind of a simple geometry, you can see fluorescence while you can excite it and block out the excitation. Okay? Many advantages of this geometry and what other things that you have. So feel free to ask questions if something is not obvious. All right, so this shows you a slightly different plot of the same data, just that now it's measured in OD, and you can clearly see it does not block anything here, but blocks the other wavelengths to OD6 or higher. Okay, block everything else. Only focus on what light you want. Okay, microscope bodies are actually not needed. Detectors are, however, needed. Okay, and you use highly sensitive detectors. I can go on and give a talk all about detectors, but today I'm not going to do that because I want to. Okay, so the standard names are given. These are very, very common detectors. And of course, they are very generic names. There are other variants also. So take this meeting. Okay, there's a strategy. I told you, if I have a molecule in the background, very large background, I will see nothing. I also told you, and you might have heard, that if you have many molecules together close by, they will also meet, because you won't be able to distinguish which single molecule is. So, what trick we do in many cases is, I call it the divide strategy. So you dilute the molecules, you dilute it, keep diluting till you see single molecules. And these single molecules are way separated from each other, so that you can never confuse. All right? Or what we do is we do what is called selective activation. Many molecules are present, but I turn on a small fraction of those molecules. They will emit all the light, and everything else is going to be dark. So you will only track that. You'll see some examples of this. Okay. The other trick that we use a lot is called immobilize. Why? If you have taken a molecule and let it float around in this room, air molecule is going back and forth. But even if it was a large molecule, a nanometer, then you can go back and forth in very, very fast rates. If you try to collect photons from it, yes, you will collect photons. They will spread all over this or a solution handle that you have. Okay, that will be useless one. So we take advantage of the fact that I can fix the molecule covalently sometimes to a place and then image it. And then we can measure all sorts of dynamic statistics. That's one. Sometimes we get lucky because biology has all sorts of these molecules that interact. A transcription factor will bind to the DNA. DNA is a very large polymer. DNA moves much more slowly. The transcription factor, the 3D diffusion, comes and binds to the DNA. I see it on the DNA as well. Every time else I see it as a blurred process. I can take advantage. A protein that is doing 3D diffusion, and I will see a blur, but it binds to the membrane. Again, I see it as a spot. I can track it. So we take advantage of that. Here's another set of tricks that people have used, artificial tricks to trap molecules and measure their activities. Okay. So listen to me. You make flow cells like this, and in there you can either have cells or you can have small molecules attached on the surface, or you can have a membrane. I will show you some examples of those, and you flow in your molecules of interest. It's kind of a microfluidic device, but it's very easy to assemble. You don't need to go to a fab fabrication facility. You just use some double sticky tape. Make this thing. Okay, so it's not that complicated. Okay, I told you about. Okay, what happens if I can't dilute it? Well, people have figured out ways where you can take a fluorescent molecule, turn it off with some excitation. Doesn't matter which excitation, depends on which dye you can switch excitation. But then you can turn on with another excitation. So it's kind of on and off and on and off. And I can reduce my intensity, then I can switch on only a small fraction. And then you track those. You won't be able to figure out which one you track, but as long as they are representative of the class, but measuring the difference. Okay, and the same goes for fluorescent proteins. People have shown that you can turn on a fluorescent one to dark one, and then vice versa, using again some UV illumination. Right? There's in the same cell, same molecule, one molecule was switched on, You visualize it for some period of time. This one disappeared because it photo bleached, and then another molecule has become on it in packet. It's actually moving around the cell. Fine. So people can do this even inside the cell, and people have come up with all sorts of strategies of increasing the number of colors they can use that and how many times it can do it. Right? So here is a dye, same dye, but attached to another dye, and depending on which color you use for triggering this particular one, the same dye is going to turn on. This dye is what is being visualized, but it turns on for each of these individuals. Okay. There are some other issues that happen. I kind of alluded to it, and you have heard about it before. If you have two molecules too close together, so this is the very sparsely decorated single molecules. You see these spots. This is a real image from one of our data sets. You see these spots of single molecules. If they are too far apart, I have no problems. I can tell these are molecules. Here one, here one. If they start coming close together, I have a challenge. Because their PSFs will start, PSF is the point spread function, they will start overlapping. You won't be able to distinguish this. Right? So, the diffraction limit is a problem. This is of the order of 200 nanometers for light visible range is what is mostly detected. The concentration hence can be limited. Okay, or the kinetics you can measure is limited. Why? I told you I diluted it. But let's say the reaction happens already at micromolar. I have added nanomolar. I won't see the reaction. Right? So that puts some limits on it. Also, how often can I switch it off? And how often can I switch on? I told you I can switch it on and off. This ratio will matter. Right? If you can't switch it off 1 out of 1,000 times, then you probably are not going to be able to measure it at 1,000 times. Okay, and then of course there is a lot of noise you can see from the camera itself. All this is nothing is there. This is the cleanest sample we could have made. Okay, I know that for a fact because of experience, and yet you will see a lot of background noise. Right? So this defines how many photons I collect and how much is the signal to ratio, and your ability to see the molecule as accurately as possible. In some cases, you might want to localize it and track it. There, this will matter. Okay, and as I said before, you can take advantage of the fact that there are multiple colors that are available. So, what I said about distribute, you can distribute over by diluting, but you can distribute by picking different colors for different molecules. Right? So, in that case, same molecule here has two dyes. One dye for the polymer on which all these are binding, and then different colors are for different proteins. And you can visualize them as a function of time. You can also take advantage of the fact that they are spectrally separated, and your dichroic mirror and your emission filter can allow you to separately collect different colors, and hence you can say, oh, this protein binds first, then this protein comes in, and only then this can answer those questions. Okay, so this is an example of something like this. If you know about a ribosome translation, is kind of the ribosome 30S subunit, which is binding to the RNA on which it is supposed to make the protein. The tRNA is tagged, which is necessary for bringing the amino acid, and there is another factor called the elongation factor. So initiation factor, sorry, IF2. This all three are colored differently, and what you see here is, as a function of time, the red one binds, the green one binds, they're color coded in the blue one is there, and this one. So it was already there. It didn't disappear. It actually photo bleaches, and then all of them bind one after the other. There's an orange one also. Okay. Now you don't need any bottles fitting. I see what is happening. The thief came first, then the police came. I know about that. Right? Imagination can go out the window because we get the video. Okay. All right. There's some quantification that you can do by what is called photo bleaching. I told you there's a single dye, one molecule going through these cycles, producing a ton of photons. Imagine there's a single ball. If it goes out, and assuming that it really goes up rather than dims, what do you see? You will see a single intensity was there, then boom, the light is gone. It's all dark. Okay, that's the background. If you have two lights and they randomly go out, then 2 steps. 4 lights, 4 steps. I'm measuring the intensity of a single light. Right? So I can use this to count how many molecules are there, even if they are in the same diffraction. I can't distinguish them as seen as an image because they are less than 200 nanometers complex, which is only 2 nanometers wide. Right? The nanostructure that I've fabricated in the nanofab facility, they're too close together. I can't, there's no single zeolite. I want to measure the reaction. I can't, but I can do this exercise to quantify. There are some challenges, of course. People have done this exercise, but for small numbers, this is all fine. But you need to correct for some things. Because every time you are going to label, of course, it's a chemistry reaction, right? So you can get inefficient labels. So you know the labels you can detect first, but how do you know that there is a one-to-one correspondence between the molecule of your interest and the label? That can not be 100%. You can also have a molecule before you put it on your fancy microscope that you want to image, you might have this before it, right? And there can be other kinds of issues too. But we take advantage of the fact that once we can figure out this labeling efficiency, then I can estimate for every molecule what is. So this is kind of graphically shown here. Imagine that there is a trimer complex, and I have multiple dyes on it, but because of some effect here, the same trimer complex can give me 3 photobleaching steps, 2 photobleaching steps, 1 photobleaching step, or no photobleaching steps. Okay, but we understand maths very well, and we know that this doesn't have a brain of it. It's going to follow standard mathematical distributions. So we can use binomial distributions to figure out all the different kinds of combinations that can create, and then we can solve for it. Again, these are methods that are established. So yeah, of course, just doing the biology is okay, but you also need to think about it. Okay, I'm going to skip more into this. Imagine now I can see the single molecule, but I can do more. It is going to be moving around. It will be moving in 2D, it will be moving in 1D. Okay, is where somebody has stuck the molecule onto the surface. Actually, a classic paper from Pore. So that molecule, I can collect a lot of photons. You might have heard already in the talks. I can also pinpoint its centroid value. It's almost, I can see the peak of the mountain, and the peak of the mountain, I can locate much better than the actual width. So what people do is fit mathematical functions to this, sometimes as simple as a 2D Gaussian, and that will give you the center, and that center is being tracked as somebody is moving the stage by 36 nanometer steps. And you can see the movement by 36 movements. People have shown you can do this as accurately. This was something favorite because I saw this data being taken into the lab when I was there. So I put it here. Then you can put it on a biological molecule of interest. This time the stage is not moving the molecule, it has a single dye. Where this is a motor protein, as it's moving on a filament, it is used to transport large complexes and vesicles in there for the kinesin motor. As it takes steps on the filament, you are visualizing those steps in real time. This is, of course, in vitro. People have done it inside the cell. Okay, you can not only measure the steps, you can also measure the time, the step size. Step size distributions are given here. I'm not going to go into the details of these things. If you're curious about motor proteins, tell you. But this time distribution is the time it took for it to take a step tells you about the biophysical process. So you get a lot of data, just not visualizing them. Okay. There's a slightly different experiment. This time around, it's not taking steps, or the step size is extremely small, but this is a DNA molecule that has been stretched out by fluid dynamics. Flow has been applied. It is in this direction, and the DNA stretches out because this one end is attached, the other end is free. Okay. So as it stretches out, somebody has thrown in a protein, and you can see spots only on the DNA because of the DNA. And this protein is doing what we call Brownian motion. It's moving only along the track of the DNA. If you did not know this, almost all proteins do. It has to go and find its site where it's supposed to go. Okay. So how does it do it? It finds somewhere and does a search process, 1D search process along the length of the DNA, and this is what you see. And of course, what we know is that it is doing Brownian diffusion, but 1D Brownian. Okay. Just like you would have expected. All right, now if it was not a DNA binding protein, it was a membrane binding protein, it will bind to the membrane and do a Brownian motion on that. But this time around, it will be 2D Brownian motion. So these are examples of 2D Brownian motions and different types of Brownian motions that have been kind of characterized on surfaces. They tell you something about the way it is interacting. And I'm not going to go into the details again, sorry, but the way it is interacting with the surface and the other proteins that might be there. So you're looking top down onto a membrane, and you will see some examples that I will show you. Okay, and by analyzing these trajectories with just many different ways, standard definition, so find the centroid, use that at the position of that thing, and then build distributions of how many steps it has taken. And this is a function called the mean square displacement, which I know from for Brownian motion scales linearly with the lag time that I take. So lag time is, I have taken one step is delta t is tau, lag time 2 tau is corresponding to 2 steps. The second position that I have attained, and the square of that thing will be twice as much as the time I have taken. Okay, so we know physical processes. Of course, not always this is true in biology. This is true for only very simplified systems. Most of the time, you see anomalous diffusion on surfaces of cell membranes, or even confined motion, because remember that crowded environment that's found inside the cell that ensures that every time you are going to reach an obstacle, and that helps you about how your movement happens on the surface. Okay, it can even on an active motor protein, you will see this kind of a diffusion. These are methods how you can characterize the motion. Okay. And if you have very large number of molecules, you won't have single molecule, right? If you have very large number of molecules, what you do? Well, activate the small number by the trick that I told you. Activate fluorescent protein, which only turns on a very small number of molecules. So this is the density of trajectories that have been measured. For each molecule, they have measured the diffusion coefficient, and you can see that there is a range of diffusion all the way from 10 to the power minus 2 to 1 micrometers squared per second. Okay, same molecule. I remember I told you about heterogeneity. Same molecule, just behaves differently, different parts of the membrane, or just the time when you measure. Okay, so this is on a membrane of a cell. This is of course on a bacteria. Somebody has measured diffusions, and they can distinguish based on the diffusion coefficient the different kinds of phenomena that they're doing. Some bound molecules versus free molecules, what kind of distributions you see, and what kind of diffusion coefficients you see, will tell you something about the biology. Again, the biology is not important. You can get these into details. Okay. Now, till now, I showed you where the molecule is moving around in the vicinity, right? So, of the order of 10 nanometers, 1 nanometers, and so on. But what about if I have sub-nanometer changes that I want to measure? And there is tricks for that too. So you can do what is called resonance energy transfer, or Foster's resonance energy transfer, in kind of honor of the person who kind of predicted the law for this. What you're doing here is that instead of having one dye, I told you that this is what is going to happen for one dye, right? Most of the fluorescence is going to be released when photons are going to be released when you are going down to a ground state. But if you have another dye molecule we call them the acceptors. If they are in the vicinity of this particular molecule, and only vicinity, this is a dipole-dipole interaction, so they don't need to physically touch. Okay, the energy of this photon, so instead of release, this electron, as before it going down by this pathway, can go to this and excite the electron pair. We say, okay, fine, I don't know what will happen. Well, the light that was going to be emitted by this molecule is now going to be emitted by this molecule. And they are spectrally different. So instead of seeing a green light, I see red light. But this is dependent on how close these molecules are. In fact, that probability changes at how far I am from the other molecule. So if this is very, very close, this acceptor molecule is very, very close, almost all the photons will go to this. Okay? And if they are very far apart, then literally nothing will. And I can take advantage of that. And this happens over angstrom circle. Right? So I can take advantage of and ask how much photons I'm getting from this and how many photons I'm getting from this, and I can take advantage of that to argue how close they are. They could be on the same protein, but I can measure the distance just by looking at the ratio of light coming from the eyes. Okay, and this is also I'm leaving out some details. If you haven't, if you have, just to give you an example of how this works. The setup looks very similar to the setups that you saw. You can do it with total internal reflection, but this time around, instead of detecting one signal, you are detecting two different fluorophores at the same time. That's all. That's the only trick. Sometimes to save money, we just split our camera into two and image the donor image and the acceptor image on the camera side by side. That saves me of having to buy two cameras for missions. Okay. And then we look for correspondence. This molecule. Okay, there you go. I have found my equivalent light signal from the other molecule. We correlate them in time, and you will see what it looks like. Okay. So here, from a single molecule, I have taken the red light and green light from the two dyes. Okay, and what do you notice? Well, there was some light coming, green light coming, some red light coming from those two fluorophores on the same molecule. But once in a while, the red light went way up, the green light went way down. What happened? The two molecules that were there emitting light, whatever molecule they were on, did this. Brought the two dyes closer together. More energy transfer is happening to the receptor, and that is emitting most of the light. And total light is, of course, conserved. Total energy is conserved. Right? So that's why the green one has to, of course, emit. And then, of course, it's in dynamic equilibrium. It's changing its conformation, not between two states, but this time three states. So that's the high Fred state shown here. That's the amount of efficiency of energy transfer, some intermediate level, and then also the low level. And I can watch a molecule doing yoga on its own in the cell in millisecond intervals. Okay. You can also, of course, build the distribution of all the different conformations it is taking. That gives you the equilibrium. And this equilibrium would be the same if I had all the molecules were the same, because the time average equilibrium is going to be the concentration equilibrium also. So every population of the molecules will also show you exactly the same. If I take a snapshot here at time and look at all the molecules that were there in the solution, they will also give you this Fred histogram. Many molecules are in this state, many molecules are intermediate state, and many molecules. And they will change as a function of the conditions. All right. So having told you about some methods, now I will try to tell you about what we are using these methods to study. Again, it's one of the typical examples of what we do. The idea is to show you that, yes, indeed, you can recover a lot of information by doing it. Okay? Even if you don't take away the biology of the problem or the physical interest what we have in this problem, just imagine that you can apply it to any system you are interested in, and you should be able to extract a lot of knowledge that otherwise be hard. It's not as if it's impossible, but it will be hard. Here, I can get it much more easily. The experiment is harder, but the information is very easier to interpret. Okay, so this is the system that I thought I will tell you about. It's called pore forming toxins. These toxins are a very large class of proteins that exist. Bacteria, present in your cells, it's present in yeast, many systems. Every system has. What's their job? Well, their job is to puncture holes in your cell membrane. Your cell membrane or some other cell. They have been kind of targeted in the so speciality is like this, but their basic function is this. One protein will go and bind from solution into the membrane. It has high affinity for the membrane. It will undergo some changes in conformation, and when it does, it will start kind of partnering itself with others, form a closed ring-like structure, and then it will undergo 4 more conformation change that will ensure that they can puncture a hole through the membrane. That's basically the general thing. Okay, and we are interested in understanding how we achieve this. Now, I'm running low on time, right? Okay, I'll try to skip through a lot of details and try to do this in maybe 5-10 minutes. All right. I want to look at these conformation changes. I want to look at whole pathway if I can. If I had taken this thing thrown on the cell, what would you see? The cell will just happen. That's what we see. Okay. If I want to see a tag this molecule, throw it on the cell, what will you see? You'll see lots of molecules will die. If you can see each molecule individually and look at its activity, I can see this, I can understand this in low concentrations. If I throw it in high concentrations, it participate in this. If I can quantify each of these molecules using photometric, how they are assembling, and then of course, if I can measure the current or the leakage of small molecules through this, you will be able to understand when it comes to exactly what is needed. Okay, that's what we have done. And I'm not going to be short because we are short of time. I won't be able to take you through all of them. Let me show you some key things. The reason we are interested in this is because not only it kills cells, which anytime you have a dysentery, these proteins have a role to play. Okay. Okay. But we like it because as engineers, these proteins also have remarkable in their ability to control current through them. And well, forget about controlling current. They are remarkable in their assembly, and hence, when you can measure current through them, you can also measure the blockage of current if some other molecule goes. And that is used for building these nano pore-based DNA sequences. So these are DNA sequences which are smaller than your phone, but would sequence your DNA. Yeah, the core of the technology is that. Now, so we are interested in several problems. We want to understand how this molecule kinds of goes from a water-soluble molecule to a membrane-loving molecule. That's amazing, right? One is hydrophobic, other one is hydrophilic. Right? Same molecule is showing both. Of course, there's some changes in conformation required. What triggers? What tells the molecule when to change environment? Do you change and hence you bind, or do you see and the structure, membrane is, it doesn't know the membrane is close by. It has to do something beforehand. Okay, so what does it do? And how does it decide? Right? The other kind of problem that we look at. So bacteria, though it is not drawn here to kind of match the scale of this, but this bacteria is about 1,000 times smaller than this. It is producing the protein. So the protein concentration is about 1,000 times larger. Anything, if you know about physical chemistry, the concentration is higher, the reaction will be faster. I should have formed pores on this much better than I would have formed on this. Yet the bacteria is producing it rapidly, they produce tons of it, and this one is the one which gets killed in animal. How did selective he is designed itself so that it will only kill the membranes on the right? We want to understand this whole process. How do you assemble? Right? I can decide to do 1 and 2 and 3 and 4 and 5. But this is going to be slow. I can do 1 plus 2, 2 plus 4, 2 plus 2 plus equal to 4, and so on and so forth. Really fast, but I can run out of these monomers and never complete course. Right? So this is fast, but it is inefficient. Which one do you use? We can visualize it, so we can know what part. We have done that, right? And found some of it. So, and then there are mechanisms by which it can decide, okay, first assemble, then puncture, or first puncture, then assemble. These are more academic questions, but I can answer them. Okay, so which one should I show you is the question. Let's see. I'm going to skip all this details because the details are not so critical. Okay, I'll show you this example. So first, we want to study this system, and we say, okay, let's measure what does it need to kill a cell. We use this assay where I take red blood cells or take any cells, any cells to do this. We put the protein, the toxin in there. The cell basically kind of punches holes. Osmotic stress will basically lyse the RBC, and the turbidity of the solution goes down. This is not a single molecule system, but I will tell you why we do it. The turbidity goes down as a function of time as you create lyse cells, and you can see that literally I have removed all cells in the solution. So this can happen at very, very low concept. We can do this at 1 nanometer. That's really bad for use bacteria, right? But we do this assay every time when we make changes to the, not chemical changes, but also when we tag the dye. Remember we talked about this. So that we can ensure the tagged molecule that we are studying is not very different from the molecule that was originally found. That's very important, right? You don't want to study some artifact. What we did is we removed all proteins from these cells and asked if the proteins are important for these toxins to recognize this cell. Okay, it was important because I was interested in the selectivity. Okay? I wanted to know why it does this. So I removed all, not me personally, in the lab, removed this, and then there's no change. Remove all protein doesn't matter. Okay. However, if I remove, so now the data is plotted slightly differently, but the same kinetics is being measured. Sorry. Same kinetics is being measured, but this time is only being reported, half-life. Okay, we do this measurement, but there's 2 changes. So we have done this experiment where the cells are measured by itself, or we have done a treatment with something called M-beta-C. M-beta-C is supposed to take out cholesterol from the membrane. Okay, and you will notice there is a 100-fold drop in activity. Something that was killing very nicely the cells, you remove cholesterol from the cells. Now that doesn't. And that immediately told us, oh, that's one of the critical parameters. That is how it recognizes. Why? Because I have this. Okay, but of course, how does it do it? So the first thing we do is we say, okay, fine, now that we know cholesterol is important, maybe it's just important for binding. Cholesterol is in the membrane, you have a mammalian cell, it sees cholesterol, binds better, and hence you get those cells. So we do a binding measurement, but our binding measurements are of course punctual. So the way we are doing it, we have put a tag on this protein. It is going to undergo conformation change, bind to this membrane. But remember, I told you 3D diffusion in the bulk is going to be a blur. But this, when it binds to the membrane, it does slightly slower diffusion. We can track it as a single spot. So these spots that we see are spots of the molecule. And we are flowing in the molecule in solution and measuring it. So the function of time. What we see in this movie? Well, they will progressively bind. Let me play the movie for you. So if they are binding, they are photo bleaching also to a certain extent. So we can correct for that. And they will, after binding, also do. Okay, we are tracking those single molecules. And if I count the total number of molecules that are on the surface as a function of time, I will be able to tell you the binding kinetics. And that's how it looks like. Okay, and I have done this. Sorry, I keep saying I because I think of my people as my own myself on. But yes, the students in the lab, they did this experiment. I'll tell you about them. So if you do not have cholesterol, the binding is actually quite robust. In fact, if anything, the binding went down in the presence of cholesterol. That's counterintuitive. I said cholesterol is important for it to recognize and kill, yet I am finding that the binding is actually reducing. Again, as I said, higher concentration should be higher activity. So what's going wrong? Something we are missing. So what do you do? Instead of looking at just binding, now we are asking what happens to the molecule after it binds. Okay, so this is a complicated plot because I haven't given you the background. But what we are looking for is how it is moving, how fast is it after binding. And you'll see there are times, right, 100 milliseconds after binding, 30 seconds after binding, 5 minutes after binding. What you notice is, so this side of the axis is called instantaneous square displacement. This side of the axis basically means it's moving fast. This side means it's for orienting. You see this red population right after it binds, it's moving very, very fast. But over time, the population changes and you have now fast moving molecules and some slow moving. Okay, same molecule, same membrane, but first it did something, now why? It's changing its conformation. Okay, and if you look at it in cholesterol, within 5 minutes, this whole transition happened. All the molecules are moving. Whereas here, the maximum I reach in equilibrium is only 50. Okay, and there's many ways I can show this. I'm going to skip this. And they are not happening because their intensities can be compared. I can look at what is the intensity of these fast-moving molecules and the intensity of the slow-moving molecules. Why is it important? Well, I can say, hey, they found their partner and they have already started assembling. When they have their partner, they move slowly. When they're moving alone, they're moving fast. Right? That would explain it. But I have the intensity, remember? So that intensity doesn't change, it means that the same molecule that I'm looking at. And I can do it in low concentration, lower and lower. I can tell you that it's not happening. Okay. This one shows you that same molecule switches between fast moving, really slow moving, and some intermediate level in the same region. So it's not gone to a region and found itself to be interacting with something else. Okay. So we interpret it as changes in the conformation of the protein, which changes its diffusion coefficient. So what we are measuring as a function of time as changes in diffusivity is nothing but the conformation changes in the way it is. And we can do some analysis. I'm going to skip that. This hidden Markov-based analysis that was published earlier that allows us to distinguish all these populations and their transition between these. Cut the quick story short. What we found is, remember, binding was not important. Cholesterol definitely doesn't play any role. However, this molecule, after it initially bound, it was undergoing this flipping of another region that is important for puncturing a hole. And this forward reaction is not influenced by cholesterol. It is the backward reaction, which is once it has inserted itself, the cholesterol interacts with this part of the region because it's now in the membrane and prevents it from coming out. It's reversible, everything is more visible, but it prevents it from coming. Change happens only in this rate constant, goes from about 5 times lower. That's all. What it did was now bias the reaction forward to the inserted state. That's what it is. Of course, we have found some more tricks along the way. I'm going to skip all this. Now we have very validated in many different ways. Let me show you one more example. Too much data, but not relevant. I want to just give you an example of how single molecule methods will help you. we did some molecular dynamic simulations with our collaborator, and what they showed us that the cholesterol also did something else. Cholesterol was going into a pocket between the two molecules. It was acting like as if it's a molecular glue. So what will it help in? It will help in polymerization. Fine. So we can do this measurement. This time around, we are adding more higher concentrations of the protein and watching them form complexes as a function of time. All right. This is just the intensity of a single dye, a single spot. If we take that back, which is disappearing over time. Okay. If I look at the intensity of this spot, you will see that it is kind of going quantized fashion, and you will see photosynthesis. And by counting how many photosynthesis, remember I told you about this? Counting how many photosynthesis steps, I can identify what is the number of molecules in that complex that have formed. Okay. And then I'm going to again skip over many things that we did to validate all that, but and to fix it. But I will show you this. This time around, I'm plotting the assembly reaction as a function of time, but for all the molecular complexes that this thing can form. This one forms a 12-mer ring, right? And you will notice as a function of time, I have the distribution of all of them. That's like a dream of a divisional permutation. Right? So you can see that initially they are all forming very quickly. They form monomers, dimers, trimers, tetramers, up to almost hexamer, but nothing larger. For a long period of time, it does nothing. Okay, and afterwards, very quickly, within a span of 5-10 minutes or so, it has assembled from these to these while maintaining some species here as well. But of course, the final thing is it will be structured. Okay. Now I can fit this data to all sorts of models and ask myself, which pathway is it? It is not the standard pathway that I think they're using. Mutation would have gone with the standard pathways. There's something more curious happening here, right? Yeah, so you can get this kind of data from DC. So now I will stop here and just acknowledge. Okay, I don't know how to. That's more tricky than this. Trying to skip over to the acknowledgement slide. Yes, got it. All right. So when I said I, it was the experiments who did most of the experiments. I did well. So Satya and Pradeep started these experiments in the lab. I haven't been able to show you all the beautiful work that they did. Abhijit and Amit in Ganpati's lab did the simulations and many simulations that I haven't been able to show you. Sandhya got us started with the protein work. And Aditya, Vishwesh, Asinto are the people who are making this work because I think we have some things that we can understand. I think, but we will have a very, very clear picture of all things that happen along the pathway because of our ability to this. And we will stop here and thank you.
Thanks a lot. This was the talk, I think, so absorbing.
Thank you.
Yeah. Very absorbing talk, and it's great that we're able to see use of single molecule microscopy, analysis of chemistry, and understanding chemistry. It's like understanding chemistry, molecule and step by step, which is really something. It's a dream. I think we can realize a complex system. And any one of you have any questions that you want to ask?
Okay, too much maybe. I'm happy to share some of the slides and not everything. Some of them is published, but if you guys want to go over them, feel free to do this. We'll be happy.
Yeah, Rahul, I have something to ask.
Yes, sir.
Ajay, you talk about the same molecule behave differently.
Yes.
I was just wondering why.
Yeah, so there, in some cases, when you saw dynamic changes, they relate to conformation changes. When you see two molecules have completely two different behaviors, but you have purified them to be the same protein, that's usually because they are covalent modifications that have happened. You did not realize them, but they are. They also can be bound to some small cofactor giving rise to them. We see it all the time, but people kind of put it under the rug. If you analyze those differences, you will actually give something more about it. I'm hoping that single molecule community does that.
Yes. Okay, one more question, quick, and that is, are we in a stage or new molecules where we can track chemical reactions step by step?
I think so. We have speed. Speed is not a problem. For example, I know colleagues who do protein folding, right? And that protein folding happens at microseconds and so on. But it depends on the question you want. Now, these chemical reactions that you mentioned, when chemical reactions is evoked, you think about small ligand and small molecule reaction. That is not possible because the method that I told you is fluorescence based, which means there has to be an organic dye, which is usually a complex enough molecule. So if you're asking, can I measure the photo cleavage of that particular dye? Yes. But if I want to, okay, how does sodium chloride dissociate? Okay, so it depends on the question you ask. For complex enough system, biochemical reaction, I think we can definitely do this. And in fact, this is one thing that I tell everybody: don't focus on the light, focus on the reaction. Okay? The reaction is the key. Tells you something which you can learn from. These are only methods.
So, how useful single molecule imaging is for understanding or is for of the intermediates?
I showed you that there's bulk methods are going to be only so much successful in telling you about the intermediate because you have to have prior idea about them. You have to then fit complicated models to it. Intermediates is actually one place where single molecule methods do the best, right? Because I don't need to have any assumptions. I see it as long as it is within the time scale of my measurement. That is, so if you are interested in looking for capturing, this is the way you do it. Very apt for this.
So filling up a very good blank in
understanding, yes, that's
the question. Yeah. So do you see any question on online?
the chat.
I don't know. If you just look at
Yes,
you can skip. Yeah.
Sorry, my computer is slow. And I'm running late. Okay, can somebody check if any questions?
There are no questions.
Which one?
Yeah,
More. Yeah. Which one? Chat. You can maybe no questions.
Okay.
All right.
Okay. Okay, Rahul, I think we are quite thrilled by your talk and great talk, I would say. Very observed, nobody would see. That's I know. In fact, I was very much, into what you're trying.
Yeah, I hope it was useful. So show you some new perspective. Many of you haven't heard about single molecules and their measurements. I hope it was something that tells you that there are other ways of looking at the same system. And of course, I'm biased, but I think this is a different and useful perspective. Yes, okay.
Go ahead.
Yes.
We are getting
Yes. What are those peaks? So remember that where is the reaction starting? The reaction started where the molecule came to the membrane and got attached. Right? So it is a non-equilibrium measurement in the sense that just like you trigger a chemical reaction, it's changing. Right? So what did you see there? Was initially the first thing that when it binds, it is in a configuration that is extremely floating, fast moving, because we knew that there was only one small portion that was necessary for it to bind initially. Okay, so it is able to move. It in fact inserts only on the top leaflet of it. We knew that also from beforehand and some simulations. So that's why it was fast moving. But the equilibrium condition in that membrane for that molecule is the flipping in and out with roughly equal rates. Okay, you saw that in the rate equation, the rate constant that we have. So over time, when you give it a little bit of time, what happens? It goes to the equilibrium. Right? It's like almost I'm jumping in the water, but initially when I jump into the water, I have some way I'm going to be paddling in the water, but then I start swimming, and that's what you're measuring. That is what will be maintained for the rest of the right? That's like almost the pink reaction that you see. Initially started off with a blue color, but the pink reaction is what maintains. That's what. And there are two conformations of the same protein. It's flipping in and out. Tryptophan is lower, flipped out is first.
Yeah, any other questions? Like another thing that I thought I will just go ahead without even asking it. The thing is that if people are interested, maybe visiting his lab sometime.
Yeah,
You know, maybe he can find some time to really, so that would be a good thing if you're interested.
Yeah, if you guys are interested, we will have it. You just write to us or you have the email, but our email is also available. Write to and we will organize. Fine. Okay. All right.
Yeah,
To run.
thank
I have a
yeah.
So thank you so much for
Yeah.
coming.
Okay, thank you.